Welcome dear students. In this class we are going to learn about vesicular trafficking of low density lipoprotein, or in short LDL. This is one of the example of vesicular trafficking, and in this class I will show you a very minute or very subtle molecular mechanism behind the vesicular trafficking of this LDL molecule. But before beginning, I want to summarize some facts about the vesicular trafficking. The vesicular trafficking, the name implies vesicular, means by the help of vesicles. So in my previous two videos I have told you that there are some mechanism for vesicular budding and there is some mechanism for vesicular fusion. Now the overall scenario is a protein enter inside the endoplasmic reticulum, go through this Golgi complex, and the ultimate destination of the protein is endosome, lysosome, plasma membrane and cell exterior. So that is, this process is also called secretory pathway, because the proteins can be secreted out to the cell exterior. That is called secretory pathway. Another thing is the protein, some protein can be actually acquired from the exterior of the cell from the into the inside. This is performed by the plasma membrane, a process called endocytosis. All we know, endocytic vesicles form. So LDL proteins are present outside the cell. So cell should capture the LDL by a mechanism. So at first in this class you have to understand what is LDL. LDL is low density lipoprotein, is round in shape, and this there are some properties or actually features of this LDL receptor. So I will write this. The first is this LDL, low density lipoprotein, is made up of monolayer of phospholipid. We know that plasma membrane is made up of bilayer of phospholipid, but LDL, low density lipoprotein, is made up of monolayer of phospholipid. This phospholipid can have cholesterol. So this LDL have cholesterol. Not only that, inside the low density lipoprotein there could be cholesterol ester. This is the actually thing. That means it is actually storage site, storage region of this cholesterol. So cholesterol ester are present. Another is this LDL is actually wrapped by a protein called apolipoprotein B, or in short we can call ApoB, apolipoprotein B or ApoB. This protein actually wrapped around the low density lipoprotein. So this is ApoB. Okay, so this low density lipoprotein present exterior of the cell. So now let me draw a cell so that you can understand. So suppose this is a cell. So this is the plasma membrane, this is the nucleus, okay, and okay, now let me draw a LDL. So if this is the LDL, then it have apolipo B protein that wrapped around this molecule, and for the LDL protein there is a receptor in the plasma membrane called LDL receptor, and this LDL receptor has two things. So I will draw in two different color, the first in red and the second I will draw in green. So this is called LDL receptor and this is LDL. Now if I draw the receptor, because the whole mechanism of this transport of this LDL actually depends on this receptor. So let me draw this receptor in here. So this is the receptor, one part of the receptor. Another part, this green part, if I draw, it will look like this. So this receptor, this region called cytosolic region, because it is inside the cytosol. This cytosolic regions have a 4 amino acid sequence called NPXY signal sequence that helps in the insertion or the endocytosis of this total complex receptor along with the LDL. This is called NPXY signal sequence. I will tell you what is the significance of this signal sequence. but at first I want to share all the information about the receptors. So this is the NPXY. Then this region, this whole region is called beta propeller. This region also have very important function, beta propeller domain. The next is called arm. So this region is called arm. This is the arm region, by which it actually attached with this apolipoprotein. Now I have drawn here 7 circles. That means these are nothing but some amino acids. Specifically we call it R1 to R7. That means 7 amino acids are present. They are cysteine amino acids. Okay, they can bind. Now when this LDL binds with this receptor, this NPXY signals actually attach with a molecule we call adapter protein 2. A protein called adapter protein AP2 then bind with this NPXY signal sequence. In my previous classes of this vesicular trafficking, I have mentioned that there are different type of coat proteins are present, like COP1, COP2 and clathrin. In my vesicular trafficking vesicle budding class lecture, I have mentioned that. So when from the plasma membrane to the inside, when endocytosis occur, clathrin protein actually responsible. So here AP2 is the adapter protein for the clathrin, and then clathrin protein actually coat all this complex and helps in the formation of endocytic vesicles. So now let's suppose these endocytic vesicles enter inside the cell. Now the endocytic vesicles is actually wrapped by clathrin protein. So this is the clathrin. So the clathrin protein is coat the vesicles called endocytic vesicles, and there is AP protein, and also these receptors also be present here. So I am writing here. Let's suppose this is, sorry, it will be inside. So this is the green portion, this is the red portion of the receptor, and let's suppose this is the LDL that it has attached with it. Okay, now when there is clathrin protein attached with this vesicle, we can call it endocytic vesicles. But when the clathrin proteins are removed, because to fuse these vesicles, this clathrin should be removed. When the clathrin proteins are removed, when there is no clathrin, no AP2, we call it early endosome. This early endosome then fuse. I have made a separate video on fusion. They fuse with another vesicles, we call it late endosome. The pH of the late endosome is approximately 5, and the pH of the outside of the cell is approximately 7. This pH is very important for the functioning of this receptor. How it will work, I will show you. So what happens at the pH of approximately 7, that means when this receptor present outside the cell, this beta propeller domain have histidine amino acids. It has histidine amino acids. Okay, at the pH, this arm is actually open in conformation so that it can bind with a LDL protein. Okay, let's suppose this is the LDL protein. This blue color is LDL. So it can bind with the LDL. Okay, but at the pH 7, when the pH falls, at the pH 5 in the endosome, late endosome, the conformation of this receptor changes. How it changes, I will show you. These portions, this red area, in that beta propeller region, in this region, the histidine amino acids that are present becomes protonated, becomes plus charged, positively charged at the low pH. At the low pH, the histidine becomes protonated, and these negatively charged cysteine molecules then attach with this positive charged molecules. So the arms are not now open, it is closed in conformation, so that these LDL proteins are released. These LDL are now released. That's why at the pH of 7 this receptor can attach with the LDL. At the pH of 5 it actually remove this LDL, and ultimately this late endosome fuse with another subcompartment we call lysosome, and then this LDL are actually digested by these lysosomal hydrolase enzymes, and all the stored this cholesterol are actually used, and ultimately these receptors that present in the endosomes or lysosomes are recycled back to the plasma membrane. And the same thing occurs when it is present in the lysosome or late endosome. The pH is low, so the arm is closed in conformations. But when it reaches to the plasma membrane, the arms become open so that it can attach with another LDL. As I have mentioned, that I always told my students that cells are very clever, so they always try to use a protein repeatedly, so it will use these LDL receptors another time. So this is all about vesicular trafficking of LDL receptor. Hope this video will help you. Thank you for watching, and always keep learning.